In this episode we are going to look at repeated dose organ toxicity. We will begin with a definition of repeated dose organ toxicity from the Alltox website. Afterwards we will hear from veterinarian Menache and briefly from Kathy Archibald, the director of the Safer Medicines campaign. This will be followed by a brief comment from Dr. Stephen Minger of King's College London, who expresses his view that stem cells could hold the key to accurate human toxicity tests. Then, with specific relevance to organ toxicity, we will see Dr. Albert Lee's company video on in vitro solutions to organ toxicity. After the video, we will look at a paper Dr. Albert Lee wrote for the scientific journal ATLA entitled The Use of the Integrated Discrete Multiple Organ Co-Culture System for the Evaluation of Multiple Organ Toxicity. Penultimately, we're going to hear from Dr. Bob Coleman, who is the Senior Scientific Consultant to Asteran plc. Dr. Bob Coleman is also a scientific advisor to the Safer Medicines campaign. Lastly, I'm going to look at whether there is sufficient human tissue available for scientists to research on. This is the first of 8 slides I'm going to look at from the Alltox website about repeated dose organ toxicity. This first slide reads, repeated dose toxicity testing using oral administration of a test substance in rodents for 28 and 90 days is used to evaluate chronic toxic effects, primarily effects on various organ systems, and to establish a no observed effect level. Depending on the potential route of human exposure to the substance, similar testing using dermal and inhalation dosing may also be assessed. Doses are selected to be sublethal but still cause toxic effects. Long-term chronic toxicity studies with a minimum duration of 12 months are sometimes required. This second slide just explains the animal tests in a little more detail. It reads, chronic toxicity testing consists of oral, dermal and inhalation subacute repeated dose studies, 28-day and subchronic repeated dose studies 90-day in rodents. Testing on both sexes is required. Some agencies may also require testing in a non-rodent species, typically dogs or non-human primates, and some agencies require longer testing periods, 52 weeks for example. The most commonly performed studies are the 28-day and 90-day oral toxicity tests in rodents. The endpoints for repeat dose testing consist of an evaluation of clinical observations, blood analysis, whole body gross necropsy, and microscopic examination of all organs and tissues, histopathology. The target organs / systems evaluated may include liver, kidney, lung, neural, that's the central nervous system, reproductive organs, and the, how do you pronounce this, the hematopoietic system, which is to do with the blood and the bone marrow probably, the immune system and the endocrine system, which is to do with hormones. This slide is about the regulatory requirements and test guidelines. At the bottom you can see the 6 OECD tests that are accepted. First one, repeat dose 28-day oral toxicity study in rodents. Second one, repeated dose 90-day oral toxicity study in rodents. Third one, repeated dose dermal toxicity, 21 and 28-day study. Subchronic dermal toxicity, 90-day study. Repeated dose inhalation toxicity, 28-day or 14-day study. And the last one, subchronic inhalation toxicity, 90-day study. This slide explains the non-animal alternative methods and where we are with them at the moment. It reads, expert working groups have noticed that interspecies differences limit the usefulness of animal studies for predicting long-term target organ and target system effects in humans, and that in vitro approaches often provide more relevant information for hazard assessment than the animal tests. Therefore, efforts to replace animal chronic toxicity models have focused on the development of human cell-based models. In 1998, a scientific working group assessed alternative methods for organ toxicity testing and made the following recommendations to promote the development and use of in vitro cultural systems for organ toxicity testing. Their recommendations were the need for better access to and techniques for preservation of high-quality human tissues for the use/development of in vitro cultures, the availability of reference test substances for each organ system, the availability of in vivo toxicity data, including human data, and finally, research to identify and validate biologically relevant toxicity endpoints for each organ system. This slide pinpoints a reoccurring theme all the way through this piece, i.e. the difficulty of doing long-term studies on in vitro cultured cell lines. It reads, the use of cultured cell models for chronic toxicity testing presents the challenge of maintaining and exposing the cells for the extended periods of time required to assess chronic toxic effects. Additional challenges for cell-based methods include the identification and use of cell culture conditions that retain the fundamental functions of the cell, tissue, organ type. This slide just explains a little bit how the in vitro models are developed. It reads, in vitro models are developed using cells or tissues from the organs that are the typical targets of toxicity. The liver is the primary site for the metabolism of many chemicals and drugs by the body, and is also the primary site of potential toxic injury, hepatotoxicity. Like all organs, the liver is composed of various cell types, predominantly hepatocytes. Liver cells contain phase 1 and phase 2 drug metabolizing enzymes, which convert many chemicals and drugs to other forms that the body can readily use or excrete. This is called biotransformation. In some cases, the biotransformed chemical is the one that causes the toxicity to the liver or to another organ. That is a metabolite. It goes on. Lee, which we'll be hearing from later on, Lee et al. Developed a system to co-culture hepatocytes with cells from other major organ systems for the concurrent evaluation of metabolism and toxicity. Species differences in the activities of the liver's drug metabolizing enzymes are one of the major contributors to the species differences observed in the toxicity of chemicals and drugs. Therefore, a human liver model for the prediction of human toxicity is particularly important. A major obstacle in using in vitro liver cells/models for chronic toxicity testing has been the limited time the cells maintain their normal functions in vitro. This penultimate slide talks about another organ rather than the liver. This is the kidney. It reads, the kidney is another organ susceptible to chemical-induced toxicity. Like the liver, perfused whole kidney and kidney slices have been used to test chemicals for short periods of time. Kidney tubal cell cultures and cell lines have been developed, and some can be maintained for extended periods. Oh, well, that's good. Likewise, cells from other human organs, such as lung and brain, as well as blood and immune cells, have been cultured for toxicity testing, which is also good, but it all comes down to how long they can maintain their function and integrity in vitro. Here we are then at the last all-tox slide. It reads validation and acceptance of non-animal alternative methods. As I said in other episodes, the ICCVAM is the American Center for Validating Alternative Methods, and the ECVAM is the European Centre for Validating Alternative Methods. And neither of them have validated any non-animal methods for assessing chronic toxicity endpoints or repeated exposure target organ toxicity. So presently, no alternative test exists for assessing repeated dose organ toxicity.
The problem with animal testing is that you cannot reliably apply the data from one species to another. As a veterinary surgeon, I take a completely different approach to a cat and to a dog. In fact, we know today how different people react to the same drug. The drug Vioxx is a good example to illustrate species differences. Vioxx has caused thousands of heart attacks in human beings. The lawyers who defended the manufacture of Vioxx in court told the judges that Vioxx was completely safe in mice. In fact, it was even cardioprotective in mice. In other words, it protects the heart in mice. But in people, it had devastating effects. The non-animal methods are basically human-based research. If we are studying human beings, then surely we should look at human cells to begin with, and not rat cells or dog cells. If it's human medicine, let's look at human beings, beginning with human cells. Okay, there are about 200 different cell types in the human body. It is possible today to obtain every single human cell type in the human body. If you want brain cells, I'll get you brain cells. If you want liver cells, I'll get you liver cells. 100 grams of donated human liver, that's a piece of liver about that big, will yield 1 billion cells on which to experiment. So there's no shortage of human cells, just a shortage of common sense.
Give us an example of an example where animal testing led us astray to make a wrong decision on a drug.
Well, there are hundreds, but a good example is Gleevec, one of the best cancer treatments that we have for chronic myeloid leukemia. That was discovered in vitro in blood from leukemia patients. It was doing really well. It went into animal tests, as all drugs are legally required to do. It was very poisonous to dogs, and so it went in the dustbin, and it was only rescued from the dustbin by somebody who'd been involved in the earlier development.
Okay.
It's now one of the best leukemia treatments that we have.
But the basic research is already starting to suggest new ways of tackling old problems. Stephen Minger can see, for example, how our ability to generate limitless amounts of specific human tissue would be of great interest to the pharmaceutical industry.
One of the major recommendations that came out of the Chancellor's recent report, what we commonly call the Patterson Report, recommendation 1 is that British academic groups, in collaboration with large pharmaceutical companies and the British government, should form a partnership to create specific types of human cells for drug screening and toxicology. Currently, Big Pharma relies either on tumor lines, which are of human origin but are cancerous cells, or they test drugs on animal cells.
So, Stephen, are you saying that this would be a sensitive tool for measuring the potential toxicological effects, the side effects of drugs that would circumvent the need for animal testing?
Absolutely. Human and animal cells are very different. They have different physiologies. The proteins that are involved in complex biological procedures are very different. And so you want to be able to use human cells. And I think we'll see Big Pharma ever increasingly embrace this idea. They may not be interested in cell therapy, because that's not really what Big Pharma is good at, but they are very interested in the idea of generating human embryonic stem cells that can be differentiated into specific types of human cells for toxicology and for drug screening.
One immediate application of stem cell research, then, is in testing the efficacy and side effects of new drugs without incurring the wrath of animal activists.
In Vitro ADMET Laboratories seeks to enhance drug development efficiency by providing state-of-the-art contract research services. iVal is equipped to provide in vitro services for the evaluation of various drug properties, including absorption, metabolic stability, drug metabolism, drug-drug interactions, and drug toxicity. iVal proudly introduces the IDMOC, Integrated Multiple Organ Culture Plate. Preclinical safety studies using animals have been limited by species-to-species differences such as metabolism, target cell sensitivity, and toxic mechanisms. Animal testing may over- or underestimate drug toxicity in humans. The FDA requires proof that lab animals would produce human metabolites before human research can be approved. Enter the Integrated Multiple Organ Culture Plate. The IDMOC supports key components of in vitro evaluation of human toxicants, human-specific metabolism, human target cells, and predictive endpoints. The IDMOC is an ideal system for human toxicity evaluation. Primary cultures from non-hepatic target organs cannot be used universally for the evaluation of drug toxicity due to the absence of hepatic drug metabolism. Multiple organ interactions can be key to drug toxicity. By placing multiple organ tissues in a shared fluid, the IDMOC creates an ideal environment for human toxicity evaluation. The IDMOC can be used in configurations of up to 96 wells. The IDMOC is truly an ideal system for human toxicity evaluation.
This is the first page of Dr. Albert Lee's article on the use of the Integrated Discrete Multiple Organ Co-Culture System for the evaluation of multiple organ toxicity. Let's begin by looking at his summary. The first sentence reads, "The application of the Integrated Discrete Multiple Organ Co-Culture System in the evaluation of organ-specific toxicity is reviewed." And of course it has to be borne in mind that Dr. Albert Lee is reviewing his own product here. Anyway, the next sentence reads, "In vitro approaches to predict in vivo toxicity have met with limited success, mainly because of the complexity of in vivo toxic responses. In vivo properties that are not well represented in vitro include organ-specific responses, multiple organ metabolism, and multiple organ interactions. The IDMOC system has been developed to address these deficiencies. The system uses a wells within a well concept for the co-culturing of cells or tissue slices from different organs as physically separated discrete entities in the smaller inner wells. These inner wells are nevertheless integrated in brackets, by overlying culture medium in the large outer containing well. The IDMOC system thereby models the in vivo situation, in which multiple organs are physically separated but interconnected by the systemic circulation, permitting multiple organ interactions." And the last sentence reads, "The IDMOC system, with either cells or tissue slices from multiple organs, can be used to evaluate cell-type specific or organ-specific toxicity." Dr. Lee's introduction reads, "Experimental approaches for the evaluation of in vitro toxicity include the use of tissue slices, primary cells, and cell lines. They are generally viewed as useful for initial toxicity screening and for the mechanistic evaluation of toxic effects observed in vivo." Dr. Lee in this part of his paper outlines what is important if the in vitro models are going to mimic what happens in the in vivo situation. It reads, "We consider the following properties to be important if in vitro experimental systems are to successfully model in vivo xenobiotic toxicity. These include xenobiotic metabolism by key organs, target cells representative of key organs, multiple organ interactions, and predictive endpoints." The IDMOC system was developed as an in vitro experimental system to model these critical properties. This unique system is based on the concept that in the animal or human body there are multiple organs that are physically separated but are interconnected by the systemic circulation. This part of Dr. Lee's paper is entitled Discussion. It reads, "While it is well recognized that, because of species differences, Results with animals do not always predict human effects. In vivo toxicity evaluation with whole animals continues to be the gold standard of toxicity testing. In vitro experimental systems, while making tremendous strides toward the replacement of animals in the evaluation of well-categorized events, such as drug metabolism and drug-drug interactions, are still considered to be inadequate for the accurate definition of xenobiotic toxicity. This slide describes how Dr. Lee's IDMOC system overcomes difficulties encountered when using standard in vitro tests for predicting organ-specific toxicity. It reads, issues with in vitro testing include the continual reliance on continuous transformed cell lines such as 3T3 cells and HepG2 cells. These cells can be used to detect general toxic agents, e.g. cyanide, strong acids and bases, but would be of little use for toxicity that, due to xenobiotic metabolizing enzyme activities or organ-specific biochemical pathways, would lead to organ-specific toxicity. Furthermore, toxicity to an organ can be caused by metabolites produced by a different organ. Such organ-organ interactions occur in vivo, but are not modelled in routine in vitro experimental systems. The IDMOC system was developed to overcome these deficiencies. It incorporates hepatic metabolism via the use of hepatocytes or liver slices, hepatic and non-hepatic target cells via the co-culturing of hepatocytes or liver slices with key cell types or tissue slices from major organs, and multiple organ interactions via an overlying medium. I decided not to read out this slide. I mean, the first sentence reads, there are a number of challenges associated with the use of the IDMOC system in the evaluation of organ-specific toxicity. And it goes on to talk about primary cells. However, if you look at the bottom right of this slide, it says organ slices. I'm going to start reading this out, and it goes on to the last slide because there's something in the last slide that I think is important. So we're going to go on to the next slide in a second, but just to read the last two lines of this slide. Organ slices. The use of tissue slices offers advantages over the use of primary cells, as the multiple cell types of the organ are fully represented. The paragraph continues, the limitations of tissue slices are that, while the cells on the outside regions of the slices are in direct contact with the medium, the cells inside the slices will be exposed to the toxicant in the medium via diffusion, which may be limiting. This need for diffusion is an in vitro artifact due to the lack of blood circulation as it occurs in vivo. Furthermore, tissue slices in the IDMOC system are as yet only viable for approximately 24 hours, and as such do not permit long-term studies to be performed. Another challenge in the use of tissue slices in the IDMO system is that, while tissue slices from multiple organs can be routinely prepared from laboratory animals, it is highly improbable that multiple human organs can be made available for experimentation on the same day, unless they are from posthumous research donation, when they are subject to the priority acquisition of organs for transplantation purposes. So the problem is evident here. It has to be remembered that this episode of toxicity test is called repeated organ toxicity. Repeated dose organ toxicity, sorry. So something that can be only kept viable for 24 hours is not going to be any good for a repeated, 28-day study, for instance. The other thing is, posthumous means like after death. So organs that come available after death, say through a motorcycle accident, they are not going to be available. They're going to be available, Dr. Lee says, for transplantation purposes rather than research purposes. So that's another problem. obtaining the organs that are needed for this long-term research. In the last sentence of this paragraph, Dr. Lee says there is an answer, but the science is not there yet. It reads, "The cryopreservation of human tissue slices, if successful, would greatly enhance the use of the human organ slices in the IDMOC system." I wonder if the following press release might hold the answer as well.
American scientists say they've successfully grown miniature human livers in the laboratory for the first time. The research teams say they hope that once the organs are transplanted, they will function normally and continue to develop. The British Liver Trust said they had no doubt this was a major breakthrough for patients with hepatitis C.
Okay, we're back to the slide before that news flash. I'm just going to read the last paragraph out. This is the last paragraph of Dr. Albert Lee's paper we're going to look at, and it throws up another problem. It reads, "Pharmacokinetics. Although in IDMOC models multiple organs are connected by the systemic circulation, there is no actual directed flow of the overlying medium from one organ to another. Therefore, the system cannot model the sequential events of drug absorption, e.g. bolus dose to the liver, followed by," and then it carries on. So the problem is here, I think in future we will be having to look at getting a directed flow of the overlying medium. That is, if we are going to get in vitro models that accurately mimic what goes on in vivo, i.e. in the human body. But for now, if these in vitro models are more accurate, well, than nothing, because basically animals mislead rather than leading us to the right decision, then I think we should carry on with the in vitro models that we got, the best ones that we got, which we can compare to known data. Forget about the animal models, but in the future it is obviously going to be a need for us to move on to more and more complex in vitro models. Okay, let's push on to the next part of this piece on repeated dose organ toxicity. In one way or another, the rest of this episode is about the availability of human tissue for research. Firstly, we're going to hear from Dr. Bob Coleman and his boss of Astorand plc.
Complained that we're using 20th century tools to develop 21st century medicines. Fortunately, there are many new technologies and companies addressing this problem by focusing on the way that the human body responds to new drugs before actually testing them out on people in phase 1, 2 and 3 clinical trials. Randall Charlton is the founder of Astorand, the world's leading human tissue company.
I think it's self-evident that mice are not men, and in order to develop drugs with more confidence wherever possible, one should use the human model, the species in which the drug is eventually going to be given. We operate a network of hospitals around the world in about 15 countries, and we collect under strict ethical conditions tiny amounts of human tissue backed by large amounts of data, all properly anonymized to protect patient and donor identity. Perhaps I should introduce again my colleague Bob Coleman, who has had more experience than pretty much any other scientist I can think of in working with human tissue.
It's important to remember that the pharmaceutical industry is developing drugs for use in humans, and for that reason a focus on human biology I believe is very important. Now historically the industry has relied very much on the use of animals, both isolated tissues and whole animals, but it's also had its problems. I've got many years experience in the industry, and I worked for 30 years in a major pharmaceutical company where the use of animal tissues was the norm. And the thing that frustrated me there was the lack of predictivity of many of the data that we generated. And even at that time, I made as much use as I could of human tissue in trying to focus on what I thought was relevant.
How are things developing in terms of setting up like cell banks, tissue banks, organ banks, which can be used by researchers? How is that kind of alternative approach progressing?
Not too well. A lot of scientists tell us at the Dr. Hadwin Trust that they would prefer to use human tissues, donated human tissues, if they could, but because they can't get reliable supplies of human tissues, then they revert either to using animal tissues or animals. The problem is that there is no national infrastructure for gathering donated human tissues for research as opposed to transplant, and then distributing those to the scientists when they need them. There is a national tissue bank and it does its best, but it doesn't have full national coverage, and without a system of collection and distribution, scientists are saying we're having to fall back on using animal tissues, and a lot of them aren't that happy about it. And again, this is somewhere where the government could step in, perhaps the Department of Health could do something. We have a system for gathering and distributing tissues for transplant. It shouldn't be that difficult to do something similar for tissue donation for research. But again, that has stalled and the progress hasn't been that good.
Would that partly involve people donating their bodies to science on their death?
That's one part of it. There are small tissue banks, especially here in London, where, for example, people can donate their brains after they've died, brain banks, but they only operate at a local, very local level. So what we need are tissue banks that can distribute tissues post-mortem, but also there's a lot of surplus tissue available from living people.
From surgery?
From surgery, from people who have biopsies and most of that tissue is thrown away, from people who have breast reductions or whatever. So there should also be a system to be able to gather those tissues, because what scientists need are tissues from people with medical conditions, but they also need tissues from normal healthy people to act as a control. So there's a shortage of tissue donation. A lot of people are unaware that it is possible to donate their tissues for research purposes, and there's a lack of infrastructure to make those tissues available.
This is a page from the Safer Medicines Campaign Autumn 2010 newsletter. This first slide shows Julian Huppert MP, who tabled an EDM to promote greater human tissue availability. An EDM is expressed in a single sentence. Its purpose is to hopefully facilitate a debate on the floor of the House. EDM 380 reads that this House recognises the immense value of human tissue to medical research and the development of safe and effective medicines. Regrets that researchers, both academic and commercial, struggle to obtain the tissues they need, and calls on the government to facilitate the donation, retrieval and supply of this life-saving and life-enhancing resource through a number of measures, including the provision of the necessary infrastructure in hospitals, reducing the level of bureaucracy involved, working to increase awareness amongst the public and the health professions of the overwhelming need for tissues, and giving all patients the opportunity to donate for research surplus tissues from surgery which would otherwise be incinerated, that is, if they wish to do so. This next slide, entitled Human Tissues Working Party, refers to the establishment of a working party centred on the desire to see ethically sourced human tissues take their rightful place at the centre of medical research, drug development and safety testing. This slide refers to a debate in the House of Commons, probably not on the floor of the House though, but nonetheless in the House of Commons, which discussed greater access to human tissue for medical research. And lastly, this slide requests that you ask your MP to sign Early Day Motion 380. So that's it for this episode. I feel I've been slightly negligent with regards to staying on topic, but the need for human tissue, whether it be grown from stem cells, retrieved from operations or donated at the end of one's life, is surely going to be important to predict accurate repeated dose organ toxicity results in the future.